Good evening tonight. I'm Carol Jenkins, Executive Director of Cystic Fibrosis Research Incorporated, or CFRI, and this is the CF Discovery Series. It was developed in collaboration with Camille Wasowich, who is a nurse practitioner at the Stanford Adult Clinic, and designed to make vital CF information accessible to the CF community. Before beginning, I'd like to note that no information presented tonight is intended for a patient's diagnosis or treatment. As always, we urge you to work together with your healthcare team for your medical treatment. Tonight, we're looking forward to our presentation on understanding genetics with cystic fibrosis. Thanks to Genentech and AMED Healthcare, they are supporting this evening and funding this event that is streaming live as we speak. And their support pays for the venue, the refreshments, and the studio we've constructed here, and any other costs associated with the program. We are most grateful. Tonight's program is being filmed by David Suhu. Volunteer Eric Martin is handling the streaming portion of the production, and staff member Joanne Davis is tracking questions for our online participants. We are a team of 4. Mary Convento is also with us this evening as our reception committee. We're live at the Crown Plaza Cabana Hotel in Palo Alto, and I encourage all of you to ask questions, whether you are here in the audience or part of our online community. Hello especially to those of you who are in the hospital. We're so glad you can join us tonight. And finally, thanks to our volunteers as they come and help us with these productions and serve food and help us with our cross-infection policies. My next step is the pleasure of introducing you to Camille Wasowich. She is my partner in crime in the production of this series. Camille?
Welcome and thanks for coming tonight, and to all of our patients in-house, welcome as well. It's such a pleasure to be here. It's interesting because this is one of our completion series for the CF Discovery Series, meant for, again, the purpose of educating both patients, family members, and friends. I had the pleasure of meeting Anabel Stenzel several years ago, actually at a retreat, where I was privileged to be a part of a discussion on quality of life and trying to manage the many difficulties of cystic fibrosis and still have some kind of normal life because of the heavy requirements of all of their medications and therapies. Anabel was outspoken and direct and impressive and compassionate. She was in the process of completing a book at that time, The Power of Two, which is promoting not only cystic fibrosis but lung transplant. Anabel is currently working at Stanford at LPCH. She had her undergraduate work done at Stanford in human biology and graduate work with a master's degree in genetic counseling from Berkeley. She's been working for the past 14 years as a prenatal genetic counselor with some experience in the pediatrics. She works in the OB genetics department at LPCH. She also volunteers here for the board for CFRI and the California Transplant Donor Network. But probably most remarkable, instead of just being a patient, she has this extraordinary version of genetics that she's going to share with us tonight. So with no further ado, Anabel.
Thank you very much. In this last Discovery Series of the year, I want to thank those of you who are able to make it in the busy season of the holidays. I'm going to try to entertain you for 50 minutes or so about genetics. I know the word genetics can be sometimes intimidating to people, but I'm going to do my best to simplify the whole world of CF genetics. And thank you for that introduction. So as we all know, cystic fibrosis is a genetic disease. I think that's pretty well known to many of us living with the disorder. Keep in mind that cystic fibrosis is one of 3,000 different genetic diseases that exist. And in many ways, CF is kind of a prototype. It's a kind of a classic disease that we can learn from and that we've learned many things from for other genetic diseases as well. It's really paved the way from understanding the disease to the gene and now new therapies as a result of knowing the gene. So this is just a diagram to remind those of you who are not too familiar with the way that CF is inherited. Cystic fibrosis is what's called recessive, autosomal recessive. Autosome just basically means the gene is on a chromosome that's in men and women. So it doesn't matter what sex you are, you're going to have CF. It's not specific to a single sex. Recessive means that we all carry two genes, one from our mother and one from our father. And in CF, you have to have a gene inherited from each parent to get the disease. So you have to have the CF gene from each parent. And that's kind of a diagram. I think this is the pointer, right? So this just shows that you get, let's say that the N is the normal gene and the small one is the disease gene, and your parents can pass on one or the other. And so there's different possibilities, and when a parent both passes on the disease gene, then you have an affected child. And there's other possibilities of not having any of the CF genes or having one from one parent or one from the other parent and being a carrier, which again is asymptomatic. Keep in mind that every human being carries about 6 or 7 recessive genes. That's just part of being human. That's why we have an extra backup copy. So it's totally a matter of chance that the people who have CF, that their parents carried that gene and passed that on by accident. So why are genes important? Well, again, I'm going to try simplify this whole complex area just to kind of show you that in the world of CF, genes play an important role, but not the only role. Genes are important for setting the background of this disease, but really it's the environmental exposures, the healthcare regimen, other things that contribute to our longevity and our health. There are a lot of studies done on the genetics of CF, and right now they believe that about 50% of our CF is genetic, and about 14% is due to a common environment between siblings, and about 36% is due to a unique environment among siblings. So again, genetics plays part of a role, but not the whole thing. And then the environmental exposures are things like pollution, exposure to infections, stress, diet, gender. And interestingly, last summer at the CFRI conference, we had Dr. Calico from Johns Hopkins speak, and he's presented some research stating that hot climates, as well as secondhand smoke, play a big role in the health of, play some role, not a big role, play some role in the well-being of people with CF. So before I go on, I want to just give a little brief Bio 101 background to just give you some background about genetics. Every single part of us is made out of cells, hair cells, skin cells, bone cells, and inside those cells is the nucleus, that's the center. And the nucleus contains these little chromosomes. They look like little X's, and they contain a very condensely packed amount of DNA. And when you unravel the DNA, and I think of a chromosome kind of like a ball of yarn, we have 23 of these balls of yarn stuffed inside of each cell. If I unraveled that yarn and I pulled it out, you're going to see this really long strand of kind of like it's like a ladder that's twisted. And that's the DNA. And a portion of that DNA is a gene. And we as human beings carry about 30,000 different genes. And they're broken up. This molecule of DNA is made out of 4 different tiny molecules called base pairs. And we abbreviate them T for thymidine, A for adenine, C for cytidine, and G for guanine. So A, T, C, and G are the 4 molecules that make up all DNA and that make up every single piece of life that exists. And from that code, there are mechanisms to read that code to create a protein. So our whole bodies function based out of the proteins that are made from reading the DNA code. So again, this is just to reiterate, genes are made up of 4 bases, adenine, guanine, cytosine, and thymidine. And when you get 3 in a row, that's called a codon, and that's your code. So I think of a gene like a pearl necklace, okay? We have instructions on how to make that pearl necklace. And this is an example of like a misspelling in a gene. There's the 4 molecules, ACTG, and let's say TTT is the correct way. Well, sometimes you might be missing a T, and that's called a deletion. Sometimes you might have like a misspelling or a substitution where instead of a T, you have a G. Or you might have an insertion where instead of 3 T's in a row, there's an A inserted in there. And all of these will result in a misreading of the code, and therefore that pearl necklace will not be made properly, and you'll get some extra pearls or some missing pearls. So again, 3 base pairs or 3 T's code for an amino acid, and our whole bodies are made up of these proteins that are made out of amino acids that are coded for. So that's kind of hopefully a little bit clear there. So a mutation in the CF gene is basically a misspelling. And this is a little diagram to show that from the DNA code, we have this secondary molecule called RNA, and it reads the code by pairing up with its matching base pair. So down here, C.G.C would be complementarily matched with the G.C.C, and that codes for a protein called arginine. And so from here, you get that pearl necklace, and the code leads to the making of the protein. Each protein is a different pearl, and then you get this pearl necklace that has to fold properly to function. And this is, again, just to show you the code, that basically we have these 4 molecules. They change to U's when you go to the RNA, so that's why there's a U here. It's basically the T. So T, C, A, and G. If you read it, this is kind of complicated, but if you read it, it shows like this is the T or the U, this is the T or the U, and then over here we have T or U, and then CAG. This would code for a protein called, or an amino acid called phenylalanine. And in CF, the most common genetic mutation is where those 3 T's are missing, and therefore we don't get this phenylalanine, it's deleted, and that occurs at position 508. So deletion at position 508 results in the delta 508 mutation, which many of you have heard of. So again, this is kind of complicated, but I just want to point out that the code leads to amino acids, and when you have a misspelling in the code and you don't read it correctly, you either get the wrong amino acid that messes up the protein, or you can get this, I just want to point out these called stop codons, where the read leads to, the misspelling leads to the inability of the protein to be made at all, and it just stops. And this will be important for later on in the talk when we talk about new therapies for CF. So CF is, again, one of 30,000 genes, and this is a spread of human chromosomes. So these are all the little balls of yarn that are stuffed inside of our cells. And you have the number 1 chromosome, which is the largest. You have 1 from your mother and 1 from your father. And you have the sex chromosomes, down here, which tell us if you're going to be a male or a female. So an X and a Y would be men, and an X and an X would be girls. So the CF gene is right there on chromosome 7. Okay. And it's on when you break up a chromosome, the chromosome is broken up into 2 segments. P is the short arm and Q is the long arm. And the CF gene is right down here on 7q13. It's very kind of complicated, but it's right there on the chromosome 7. I think it's probably next to the gene for compulsive behavior and intelligence and all those wonderful features of CF too. So the gene was discovered in 1989 by lots of different researchers, specifically Dr. Francis Collins and Lapsu Tsui from Toronto, through many mechanisms that I won't go into, but it was pretty much miraculous. And from the CF gene, again, it set the stage for the discovery of genes for a lot of other diseases. So when you actually look at that pearl necklace of the CF gene, there's about 1,480 amino acids, which means that there's about 1,480 pearls, if you will. And that pearl necklace will kind of configure itself. It rolls up into a little ball in a very specific way, and it sits inside a cell membrane here and allows water and salt to pass through cells. So you can imagine if you're not coded correctly, if the DNA is not coded correctly and that protein doesn't wrap itself around in the right way, it's not going to work as well, and the water and salt won't go through the cell properly, and you'll get thick mucus. And that protein is called the Cystic Fibrosis Transmembrane Regulator, or CFTR. And like I said, it's in the cell membrane. And it forms a little tunnel, little channel. So right now, depending on what literature you read, there's about 1,500 to 1,800 different variations in the CF gene that cause the disease. And we call those variations mutations. You might also hear the word alleles. Basically, it's one gene with many different changes in that gene. So think of a giant pearl necklace and all the different beads of all the different colors and all the different ways of styling that necklace to make it supposedly functional. So currently in a laboratory, you can test for anywhere from 23 to 97 of the most common CF mutations or changes in the gene. And that accounts for about a 90% accurate test in about Northern European populations, and I'll go more into that later. And as I mentioned earlier, delta F508 is the most common mutation that occurs in about 70% of people with CF. So again, delta means deletion, F means phenylalanine at position 508. So at position 508 of the gene, there's 3 T's missing, which is supposed to code for phenylalanine, but it's not there, so there's no phenylalanine. So there's a deletion. And if you looked at some of the historic literature on CF, you can actually trace that delta F508 gene mutation all the way back 52,000 years to a common ancestor in Europe. So basically, we are all related.
Anna, there's only 23 to 97 mutations that are readily testable. How do the other people get their definite diagnosis of CF? Do they still use the sweat chloride test, or how do they know they have CF?
So the question was, if there's only 23 to 97 mutations readily testable, how do people get their diagnosis of CF? I'll go into that more in a minute, but there are more kind of intricate testing mechanisms to look deeper into the DNA beyond that 97, which not everyone does. And there's kind of a sequence of testing. Because the delta 508 is seen in almost 70%, you usually start with that one first. But then if they're not Caucasian, let's say they're not from Northern Europe, you might start with more mutations that are seen in other ethnic groups. If they still don't have that, then there's different tiers of testing, which I'll go through in a bit. But good question. And the other thing I want to mention, not only are we all commonly related, but we also know that people who are CF carriers, why did this gene exist for 50,000 years? Why is it still around now? Well, there's research to suggest that the CF carriers, people who carry one copy of the CF gene, actually have an advantage in evolution to somehow not catch diseases like cholera or typhoid or diphtheria, kind of diarrhea type of diseases. And so it's been an advantage. So it's been passed on as kind of an evolutionary thing. So in the range of the CF mutations, so that pearl necklace we're talking about, there's different classes of mutations, and it's probably hard to see from here, but basically there's 5 different classes of CF mutations. This first picture shows you the normal. So you have the DNA down here, it creates a protein, that protein gets shuffled to the cell membrane and forms a tunnel, a little channel where water and salt can go through cells, and that results in normal mucus. Well, in CF, that doesn't happen properly. In class 1 mutations, you can't even make the protein properly. It doesn't even get out of the middle of the cell. In class 2 mutations, it gets to kind of a processing area, but it never actually makes it to the cell membrane. That's the category where delta 508 is. In class 3, the protein is made from the DNA. It travels outside the center of the cell. It gets processed, gets shuttled to the edge of the cell, but then it's blocked. It doesn't work, okay? And then number 4, class 4, it gets all the way to the cell membrane, but it doesn't work properly either. So this is block and regulation. This is altered conductance, where it doesn't work in the conductive sense. The chloride channel doesn't process correctly. And then the last one is that the CFTR is made correctly, but there's not enough of it. So you might get some to the channel or to the membrane, but not enough to really prevent symptoms. So about 15% of people with CF have class 4 or 5 mutations, and those tend to be the people with the milder disease who are kind of pancreatic sufficient and maybe get diagnosed later in life. The class 1, 2, and 3 are the ones that are, like you can see here, they don't even make any protein at the cell membrane. That water and salt is not passing through cells and tend to have much more classic disease. So this is another somewhat intimidating slide about all the different mutations that are commonly tested for. So in 2001, the American College of Medical Genetics decided that we should screen every single pregnant couple, every single couple considering pregnancy, everybody related to someone with CF, for CF, to see if they're a carrier. And so they found the most common 25 mutations, and those most common ones were the ones that were found in 1 in 1,000 people or more. Okay, so they considered common as 1 in 1,000 people or more. And then in 2004, they kind of changed their mind, and they said, wait a second, these 2 other mutations that are in red, you probably can't see them, at position 1078, there's a deletion of T. Okay, so that's one that they found wasn't that common, so they took that out. And then the I148T is a mutation that really isn't a mutation unless it's co-inherited with another deletion. So that was kind of realized to be just a variation, and it's not really a mutation unless you have this other deletion with it. So they took that out. So now currently the standard of care is 23 mutations are tested for in routine carrier testing or prenatal testing. So this is a little bit intimidating. I won't go into too much detail, but I just want to point out there's something that the molecular genetics lab director at Stanford coined the word the CF arms race. And there's this belief that the more mutations you test for, the better off you'll be. And so there are people with CF who are trying to get a diagnosis of CF. Maybe they'll have a mutation test that has 23 mutations. And some of them will be positive on that test, and some of them won't, because maybe they have a rare mutation that's not part of the 23. In this crazy slide, it shows that if you tested only for the Delta 508, okay, that's the most common one, you're going to pick up about 49% of people with CF, people with CF. If you increase that to the 23 recommended mutations from the American College of Medical Genetics, you're going to pick up about 72% of people with CF. If you tested for 160 mutations, that's expanding, including rare people, so 140 more mutations that are more rare, you're going to pick up about 90% of people with CF. So in the diagnostic aspect, when you're trying to diagnose someone with CF, generally more mutations is better. Okay? However, in carrier screening or in prenatal diagnosis, we have more trouble with that, because here we are in our clinic, we see general population people with no family history of CF. They come to us just because they're pregnant, and they are offered a routine carrier test, which is the 23, and we miss some. If we increased it to testing every single random general population person for 160 mutations, we might pick up a few more, but we have to be careful about how many mutations, the cost of that. Also, there's issues of false sense of security, that some people who have a 23 mutation panel might feel more, uneasy, that they don't have as as good of a test versus somebody who has a 160 panel mutation test might feel a little bit more peace of mind. But truthfully, there are no guarantees. And we definitely have seen people who have the, very good tests who still have babies with CF. So there's kind of a fine line there.
Is this a broad question? But do many or most insurance companies actually pay for genetic testing if you do not have a history?
Yes.
Yeah, they do.
So the question was whether or not CF testing is covered by insurance. So because the world of insurance tends to work on standards of care, that means those are kind of normal practice guidelines or kind of expert opinion recommendations. And CF carrier testing is an expert recommended test since 2001. The American College of OBGYN and the American College of Medical Genetics support and endorse and encourage and put the green light for that. So therefore, insurance companies tend to follow those kind of protocols or guidelines and tend to cover it. I've rarely encountered, you can chime in if you want, I've rarely encountered any insurance that refuses to pay for it.
Let me give you an example of extreme. Like, let's say a sibling, I'm a carrier, so if a sibling of mine wanted to determine if he or she were a carrier, would they pay for that even though they wouldn't be getting pregnant and having children?
So the question is whether or not, if there was a family history of CF, would insurance pay for it? Unless you had a really bad insurance company, tends to cover it, especially if there's a family history. So, was there another question?
So, from an adult perspective, if the genome was mapped out in 1989, which led us to the ability to find out more about this CFTR gene, which led us to these tests. So, in the adult world, it's the CF32, which we do at Stanford. We move into an Ambry testing for the whole 1800. That's $3,000. This is about $178. So sometimes insurance companies will pay for the 32. Often, unless you're going to have a child or you have such significant disease, because some of our older patients have borderline sweats. And so they don't know really, do they need these extra medications that are going to cost $65,000 a year for the full therapy? And you'll go through other testing, chest CTs, to see what exactly is your extent of your disease. What is your pulmonary flow time? What is your FEV1? So it's not just one thing, but if you are being referred by a pulmonologist for further evaluation and treatment, that's one subset. If you're asking for additional information as a carrier status, they may or may not pay for that. I definitely would call. I think most people could pay the $179. $3,000 is a whole other ball wax. And you
Yes.
take that test to find out you're more covered.
Thank you, Camille. So some points were brought up that I want to mention. When there's a family history of CF and you know the CF mutations in that family, you can do what's called targeted family testing or targeted mutation testing. So let's say I have a Delta 508 and an R347H, and my brother wanted to get tested. Well, I would tell him to ask for that mutation only. And, never say never in genetics, but for the most part, that would rule in or rule out whether or not he's a carrier. Also, the other thing brought up was something called sequencing. And what sequencing is where you take that pearl necklace and you actually look at each pearl under a microscope very carefully and look at the color and the consistency and the shine and the value of each pearl, and it's a very laborious process where you sequence the gene and you can actually read the entire code, and that's why it's $3,000, and it's mostly used for people for diagnostic purposes. Let's say they do one of these panels and they don't have a gene found or mutation found, then they might go to further testing like that. So.
One more question on the last
Sure.
slide.
Sorry.
You didn't mention the center segment there.
Yeah.
That-
It's so complicated.
It's probably not worth the time.
Yeah, it's just, I tried to, when you talk about number of alleles versus number of people, I tend to go more for the people explanation. But it's basically saying that if you tested only for Delta 508, you would pick up 70% of the mutations out there. which is what I had mentioned earlier, that 70% of people carry the Delta 508. If you did the 23 mutations from the American College of Medical Genetics, you're going to pick up 85% of the mutations out there. So, hope that makes sense. So ethnicity does matter, okay? This is a disease that originated in Northern Europe, but as you know, populations migrate, and we have seen CF all over the world. It's less common in people of color, but it is definitely seen. People of a non-Caucasian background tend to carry genetic mutations that are a little bit rare and undocumented. So here we have Northern Europe. About 1 in 2,500 people has it in Northern Europe, and the carrier frequency, if you did the math, is about 1 in 25. That means that 4% of all Caucasians walking to this earth are CF carriers. That's a lot. That's a lot compared to some of the other rare genetic diseases we see. And then if you looked at a general genetic test, 23 mutations or 97 mutations, you'll generally pick up about 90% of those carriers. So you're going to lose about 10%. 10% of Caucasian people walking this earth who are CF carriers have some very rare mutation that's not going to be picked up on a standard test. Southern Europe also similar. Ashkenazi Jewish, these are the lucky ones because they got the highest detection rate. I shouldn't say they're lucky, but they have the best pickup rate on a genetic test, and that's because they have 2 mutations that are very well described in Ashkenazi Jewish populations. One is W1282X. Forgot to mention earlier that when you see an X at the end of your mutation, that means that it's a substitution for any other amino acid. And it generally is a stop codon. That means it generally just stops reading that sentence. It just breaks off right in the middle of the sentence. That pearl necklace just gets chopped off, and it just doesn't work well. So these tend not to be great mutations. And then also this one has been described in Ashkenazi Jewish people as well. And then as you go down here, you see Hispanics, African Americans, Asians. You do see it in those populations. The carrier frequency is much less. And the pickup rate for the tests are not as good. Again, if you're using a 23 mutation panel, 97 mutation panel, you're going to probably miss some of those really rare mutations. And people of these backgrounds tend to have really rare mutations. So I do want to mention that there's a huge area of research called looking into non-classic CF, or what they call now CFTR-related disease, or the spectrum of diseases. And I want to focus kind of on this diagram down here. This line here shows that for you to be a carrier, all carriers have 50% CF, basically, because you have one gene that works and one gene that's the CF gene. But carriers are healthy in general. Once you start losing CFTR function, either because of a different mutation or a second mutation, or the class of mutation you have, then you start getting into symptoms. So there's this gray area here where there are people who kind of sort of have CF, but they have it on a very small level. I think of it like turning on a faucet. Some people have it dripping. Some people have just a deluge and a flood. So it depends on how you look at CF that way. And some of the symptoms that these individuals can have are pancreatitis, congenital bilateral absence of the vas deferens. That's a mouthful. We call it CBAVD. And basically the vas deferens is the tube that allows semen to exit the body. And that's a very sensitive tube to mucus clogging. And so men who have just a little bit less CFTR function tend to have that vas deferens clogged and not be fertile. And this is a certain mutation. Bronchiectasis, allergic bronchopulmonary aspergillosis, chronic sinusitis. These are just some examples of the diseases where they've done research in these groups and found that a significant number of people with these problems have CFTR mutations. So this red didn't come out very well, but I always get asked in genetic counseling, especially for a pregnant woman or a carrier couple, what will my child's CF be like, based on these two mutations? And the bottom line is the genotype cannot completely predict the phenotype. Those are kind of genetic babble words, but basically it means your mutations don't necessarily predict how you're going to do with the disease. There's so many environmental components. There's so many other factors, other genes in our body that play a role. So, people tend to call the Delta 508 a severe mutation. I'm so sorry, your kid's going to have classic CF. They're going to be pancreatic insufficient. They're going to have to take enzymes for life. Probably going to have some lung involvement, or yes, they're going to have lung involvement, but that does not mean they're not going to live till they're 50 or 60, because we see that, because environmental influences are so very strong. There was one study of a man who looked at 114 long-term survivors. These are people who were diagnosed young and survived old. And they didn't find differences in their genotype or their clinical variables. So he questioned, does genotype really make a difference? We all know that attitude, lifestyle, parental support, socioeconomics, all of those play a huge role. So we always caution how we counsel people based on genotype. So I'm going to talk a little bit about pancreatic function. You have to have less than 2% of CFTR function, meaning you have to have less than 2% of that CF protein working to be pancreatic insufficient. That means you need enzymes for life. If you have more than 5% of the protein working, then you'll be pancreatic sufficient, which means that you won't need enzymes and you'll be able to digest food. And this tends to be the class 4 and 5 mutations. For lung disease, it's again we're less able to predict lung disease based on the genotype, but there are some milder mutations that can help. And then there are some studies looking at Delta 508 that you're more likely to get susceptible to Pseudomonas, those kind of correlations as well. So there's the background of your CF genes makes a difference. There's some people who have 3 CF mutations. They tend to be more severe with their disease. So you have 1 CF mutation on 1 chromosome and you have 2 on the other chromosome. And we see that now and then. And then this I-148T I mentioned already, that's the one that was taken off the ACMG panel because it only is a real mutation if it's co-inherited with a specific deletion, a deletion at position 3199. There should be something else there, sorry. So again, it's not really a deletion, not really a mutation unless it's co-inherited. So there's a lot of variations in the CF gene. Without getting too complicated about it, I just want to say that it's not just a pearl necklace. There are little metal pearls in between each pearl. And those metal pearls play a role. And some of those metal pearls are chopped out of the gene, and sometimes there's mutations in the metal part of the necklace that influence the pearl next to it. So it's very complicated. There can be different variations in the CF gene, what we call polymorphisms, different shapes of the gene, that some don't even cause any disease. Many of us are carrying deletions and mutations in all of our parts of our DNA that don't cause any problems. And we've seen over 200 variants in the CFTR gene. And then I'm going to give you an example of one that's all over the medical literature of this particular mutation, R117H. So at position 117, there's a misspelling. Instead of an arginine, a histidine is there instead. So this is, again, it's a misspelling leading to the wrong amino acid. But it's really not a mutation unless it's co-inherited with this variant. This is, again, a little intimidating, but bear with me here. Everyone still alive? Okay, so here we have the CFTR gene. Exons are the part of the gene that stay with us. That's the pearls. In between the little lines here, we call introns. That's the little metal parts between the pearls. I'm wearing this necklace for a reason. It's got purple beads and it's got little metal things in between it. Can't probably see that, but the point is that this area here, it gets chopped out. Okay, it doesn't really play a role in the final making of that protein. But sometimes in this portion between exon 8 and 9, there's a portion of T's, thymidines. Sometimes there's 5, sometimes there's 7, sometimes there's 9. And depending on how many Ts you have, that's going to influence how the processing of the final making of this DNA will happen. If you have 5 Ts, the chopping of the 8 and 9 don't work as well, and you get a little bit less functional CFTR. In fact, you only get 5%. If you have a 7T or a 9T, meaning what I've written up here is 7, you're pretty much okay. It splices fine, it processes fine, and your exon 8 and 9 are going to work. And so you have 85 to 10% function, which is enough. To make things even more complicated, just down the road from here, there's a whole segment of TGs. Well, the number of TGs you have then influences the number of Ts. So now, if you have the short version, the 5T, and then you have a 13 TG repeats, you kind of get a double whammy. And unfortunately, the splicing or the processing of the exon 8, 9, and 10 don't work as well. So that explains why some people with CF, family A with delta 508, delta 508, family B with delta 508, delta 508, this family's disease might be much worse. This family's disease might be okay. Well, maybe there's some TGs and 5Ts playing a role here. And that's just one example of how variants in the gene can actually play a role in how that is processed. So this is a troublesome slide, so I'm going to kind of talk more than say anything. We had a little technical difficulty. The biggest question that many people ask is, why do we see so much variation in families with CF? So you can have, within families, so you can have a mom and dad with this two CF mutations. They pass those on to their child. You might have a boy with CF and a girl with CF, and somehow one gets hospitalized so much and the other is totally fine out playing soccer, lives till he's 50, whatever. You have such variation. Why is that? Well, siblings share 50% of their genes, okay? That's just what happens when you pass on the chromosomes and making a baby. And then environmental components play a big role, gender as well. So there is definitely There's definitely things that play a role. One of the big hot topics in genetics for CF right now is modifier genes. What is a modifier gene? A modifier gene is a gene outside of the CF gene that plays a role in something, nutrition, lung health, immune system, and that can influence how one's CF will be. And this is a list of a bunch of different modifier genes that have been described, and they're more and more hot on the press all the time. There might be a modifier gene for meconium ileus. That's the bowel obstruction that you observe in some people with CF on chromosome 19. On chromosome 11, there's a gene that's involved in cell differentiation under stress and inflammation. On chromosome 20, there's a gene that's implicated in weight maintenance. Well, they've recently discovered that some of those genes might play a role in modifying how severe your CF might be. And that explains why siblings might be different. Maybe Johnny and Mary got the same CF genes from their parents, but maybe one of them got the weight maintenance gene and one of them got the inflammation gene, and you see such a difference. So modifier genes are huge in how they might affect.
So it's fair to say that modifier genes could have a positive or negative influence on the outcome of a person, right?
Correct. And the other genes, the MLB1, which is, this one, mannose binding lectin 2 gene, plays a role in inflammation and the immune system. And that's been shown that people who have a certain variant of that gene might acquire Pseudomonas earlier than other kids. There's also the alpha-1 antitrypsin mutation. Alpha-1 antitrypsin is a genetic disorder that causes emphysema and liver disease in adults. I don't remember the frequency of that, but it's a disease that's pretty well known. And if you have a certain mutation on your gene for that, you might be more predisposed to liver disease if you have the CF gene too. So in fact, it's found to have a 5-fold increased risk of liver disease. And then down here, the TGF-beta, transforming growth factor gene, it plays a role in inflammation. And recently they found that kids who carry this particular mutation, a variant of this gene, when there's secondhand smoke in the home, they tend to do much poorer. So somehow this predisposes that secondhand smoke to have even more deleterious effect. So your environment, your genes, they all kind of play a role together. So why does it matter to know your CF genes? Well, for most people, they want to know, do I really have CF? If my sweat chloride test is borderline, if I don't really have a lot of symptoms, do I really have this disease? And this can be very important for people who are trying to apply for aid, for disability, for pharmaceutical financial assistance. You need to have a diagnosis. So we do different types of testing, mutational analysis I talked about, sequencing I talked about. In very rare people with CF, 2% have a complete deletion or a duplication of a part of their CF gene. So the entire gene is just extra or missing. It's a very complicated thing. And then again, the 5T and the 7T analysis and the TG analysis plays a role. Some of us can learn about our severity of CF. If we know our genotype, keep in mind you have to take that with a grain of salt because it's not completely predictive. For family members, it can be helpful to have carrier testing, or in some families who choose to know if their unborn child has CF, you can do prenatal testing. Some couples who are carriers choose to do pre-implantation genetic diagnosis. That is a type of in vitro fertilization where you take an egg and a sperm from 2 CF carriers, you create embryos, you sample one cell of that embryo and see if it has CF. And the embryos that don't have CF, you transfer into the mom. So this is a way of pre-selecting embryos, and it's kind of a modern technology that's available, but it's only available if you know the mutations in the family. And then pharmacogenetics, which I'll talk about, new therapies. I think that's the most exciting part of our genetics and why we should know our genes. I already talked about the American College of Genetics guidelines for who we should offer carrier testing to, and anyone seeking a pregnancy or currently pregnant or anyone who has a family history or has CF should be offered CF carrier testing. In prenatal diagnosis, which is where I work, we offer different tests. There's tests to check the placenta cells early in pregnancy to see if the baby's affected. There's tests to check the amniotic fluid later in pregnancy to see if the baby's affected. And then we also do non-invasive testing where you actually do an ultrasound, look at the bowel. It's definitely not a diagnostic test, but it's a way of getting a clue. And in some cases, when there's echogenic bowel, which is this little white area in the bowel, it signifies baby constipation. If you see that in a baby, there's a small chance that baby has CF. There's a greater chance that baby doesn't, but it is a clue. So there's different testing options, and I just kind of wrote down a few of the labs that do testing for CF. There are over 50 labs that do CF testing, and which lab is better, I can't comment on, and which lab you should use would be up to your physician who orders the test. But just to give you some examples, you have Genzyme, which does what we call the CF Plus, and we recommend that for people who are not Caucasian because it tests for 97 mutations. So that's the most clinically available maximum number of mutations you can get in a panel. And it has a good detection rate. It's not perfect. So we always tell people, even if you're negative, there's still a small chance that you could still carry the gene. And the cost currently is about $800. There's another lab, ARUP. They do a 32 mutation panel, 425. Sequencing, of course, is a little more. CF32 mutations at Stanford is actually, sorry, this is wrong, it's $1,595. Now, why would a test that's exact same be $425 at one lab and $1,595 at another? Depends if you want to buy your Tom's of Maine natural toothpaste at Whole Foods or if you want to buy it at Big Lots like me. So, again, I'm not going to endorse or comment on anything, but to say that there's different prices at different labs, and it depends on what your insurance or your physician recommends.
Why is there no detection rate for ARUP?
Probably because I didn't have time to look it up. I'm sorry. But they tend to be similar. They're about 90% for Caucasians. The detection rates all tend to be very similar. And the reason that says 97 for Stanford is because that's for the Ashkenazi Jewish population, where it's the highest detection. But in general, they're all about 90% for Caucasians. Ambry Genetics does the most comprehensive testing. They do what we call tiered testing. So they'll start with the Delta 508. If the patient doesn't have that, they'll go to the next level. If the patient doesn't have that, they'll go to the next level. And then, sequencing, where you go through each bead carefully, laboriously. Or if they still can't find a mutation, they'll actually look for deletions or duplications. That means chunks of DNA that are missing or extra. And they have the highest detection rate, but biggest price. And then other ones. So most important, and the reason that I think genetics is really hot right now in the CF community is because based on our knowledge 20 years ago when the CF gene was discovered, we are now getting to a place in the miraculous medical advancements of CF that we can actually use this information to help therapeutically. And the company Vertex, which I'm not a part of, but does have 2 medications that are up and coming for people with CF based on their mutation. So VX770 is going to be called Calideco. That's the new name for it, Calideco. Sounds like a furniture or something to me, I don't know. It's a potentiator. A potentiator is something that really helps the CFTR protein reach its maximum potential at the cell membrane. And so people who have G551D, that's a mutation that's seen in about 1.6% of patients, they can basically get that fixed when they take this pill. And that's huge. No more inhalations. I mean, it's huge to have an actual oral drug. You'd still have to do your regular treatments, I understand. But this is in process where they filed with the FDA and they're waiting for FDA approval because the clinical trials were so promising. The other drug that's in the works for Vertex is VX809. I'm curious what they're going to call that one. And that's what we call corrector. A corrector is something that corrects the protein not working. So an example of that would be the Delta 508 mutation. It's the Delta 508 mutation. The protein is made, but it gets out of the middle of the cell, but then it gets degraded. This corrector would help bring that to the cell surface. Then they might throw on a potentiator drug as well, like Calideco, and then help that person's CFTR work properly. So these are milestone breakthroughs that we will keep our fingers crossed that that's the next big step there. And then Adalurin is a drug that's in phase 3 clinical trials. And it's made by a company called PTC. And basically this drug has been used in other genetic diseases, and what it does is it corrects mutations that are stop codons. So if you have a mutation that ends in an X, that means that your pearl necklace is being made and then suddenly stops and it's chopped off and never makes a necklace. Well, somehow this drug allows the DNA to keep reading through that break, through that mutation, and continue reading so that your pearl necklace is not broken off. So that affects about 10% of patients. And again, we'll keep our fingers crossed that that's the next breakthrough. And then, of course, the biggest one is this Vertex V809. If that could help correct the genetic defect in 70% of people with CF, I think we may be close to going out of business, but I won't say that far yet. That's the hope, maybe. So breakthroughs are great. I do want to comment on a newer area in genetics called direct-to-consumer testing. That's something where you can actually go online and buy a genetics test with a credit card, have it shipped to your house, and then you do a little mouthwash and you swish in your mouth and then spit in a cup and send it back, and they analyze your DNA that way. You don't need any doctor, you don't need any insurance company, and this is the big up-and-coming and current thing right now. And examples of that are companies that are based, a lot of them are based in the Bay Area, not all of them though. Navigenics, 23andMe, Council. You might see the blimp every now and then with the 23andMe on it. And this is a way of testing. They offer these as multiple diseases on one particular panel. So it's not just for CF, but you would get information about all kinds of genetic disorders and things that you may not want to know about, like Alzheimer's disease or some sort of rare metabolic condition or some, cancer predisposition or heart disease predisposition. So it can be quite ethically challenging and controversial whether or not somebody should do this kind of testing in the absence of medical professionals. Some of these companies do offer genetic counseling, but some don't. So it's really, you have to be careful about what you order on the internet. And then we want to make sure that are these results really accurate? They may be negative for your CFTR gene, but what does that mean? How many mutations are they testing? What is their accuracy?
Why do you think this genetic testing is regulated by the FDA or some other organization?
It is. It's increasingly regulated. There were some issues that came up a year or two ago, and so there's much more scrutiny. All the labs have to be kind of accredited labs, so there's more and more.
You don't need a doctor's approval, you can do these independently.
Yeah. So I don't know why they can do this without a doctor. I'm not sure.
When you look at FDA regulations, you have European standards, and then you have the United States and American standards. Our standards are much higher. So some things will pass in Europe and have been utilized in Europe for years because much of Europe is socialized medicine. So when you move over here, where a lot of the research and development is being done, they have to be funded some way. So in order for you to get FDA approval, you have to go through multiple clinical trials, and there has to be this huge section to make sure that you're not hurting anybody at any time. There's standards and protocols for that. So when you look at these particular testings, it's kind of like when you decide, oh, I could have missed my period, I'm going to go to the store. But in reality, if you really think that that little plus is positive for pregnancy, you're going to go get a blood draw from your doctor. So this may lead you, it may confuse you, and it may or may not be accurate. But if there's such a desire in your heart to want to pursue this, if you do have some information that pans out being positive, you could always follow it up. But I certainly think that when you buy stuff from the television or when you buy stuff through a mail order catalog, you may or may not get what you think you're going to get. And there is no emotional contract or physical contract like there is when you have a relationship with your physician because you're in there.
Yeah, how about the protection of your medical data?
So
How's that being?
So in the terms of direct-to-consumer testing, again, I don't work for such a company, so I can't comment too much, but my understanding is that you get assigned a number, and this is through your email address. So they really don't have too much personal information but your credit card. But, there are ways to make it confidential, and that's the attraction of these direct-to-consumer companies to a lot of people who don't want this on their medical record, that they can find out if they're a carrier for something based on, without issues of insurance discrimination. So I'm just going to throw in a few last-minute genetic twists. I know we're getting kind of late. So I just want to throw out there that there is a mutation on something called mitochondrial DNA, which is way too complicated to get into, but there's other DNA besides our nuclear DNA that predisposes to hearing loss from aminoglycosides. And aminoglycosides are things like tobramycin, gentamicin. Many of us use amikacin, those antibiotics. There are about 1% to 15%, depending on what you read, people with CF who get aminoglycoside-induced hearing loss. And that can be due to overdose or wrong value levels or something, but it also can be more easily happening if you have this genetic predisposition. And so there is a test that can be done to check if you're predisposed to this. They've done a lot of studies to say, should we test everyone with CF for this aminoglycoside predisposition for hearing loss? And the consensus is pretty much no, because tobramycin and gentamicin are so valuable in fighting Pseudomonas that the risk benefits of not taking those drugs versus the small chance of losing your hearing is not good. So I don't know if you want to comment more.
So CF national guidelines requires that you have a hearing test either every year or every other year. Once you've been documented that you've actually had hearing problems associated with glycoside, typically we pull that off and don't typically give you that as a form of pseudomonal. So when you come in with a CF exacerbation, you'll get 2 drugs for pseudomonas because you want to enter the cell and then you want to kill the DNA. So it's 2 cells and 2 antibiotics in combination, one of which typically is glycoside. But if that is the case, then we would choose something else, number 1. Number 2, a lot of our patients for pseudomonal suppression, because I can't eradicate an organism once I've already grown it for many years, we will either have it on Toby or Clistin, some patients are on Caistin for suppression, and they still need to have either annual or every 2 years hearing tests. It is rare, but some of our patients do have hearing problems. Typically we dose now, there's many options, but typically we dose now based on milligrams per kilogram. We high dose it, and it's kind of like cake and icing. So you have your cake, and then you have the icing over it. When you do antibiotic coverage and you're using 2 agents for Pseudomonas, you have a high peak with tobramycin or glycoside, which is kind of like your cake, and then it comes down quickly. Why? Because the second drug you're going to give is going to push your ability to kill organisms out further. So you have a high peak, it follows by your second organism agent, and you actually maintain a higher peak level with decreased hearing and renal problems, because that is the situation. And so people now, versus like 10 years ago, have that problem much less, and we tightly regulate them. And if you've had problems with your kidneys, we'll go even to a lower dose. In order to regulate your therapeutic levels, we draw blood.
Thank you. Thank you. So again, these are things, these last 2 slides are really like rare things, but I just think they're so interesting that I had to share them. So the hearing loss gene. And then the other thing is 3 different ways that CF can be inherited that are super, super rare. But there's one thing called uniparental disomy, okay? Let's imagine that there's a kid with CF out there. They test the parents. Mom has the gene, but dad doesn't. How do you explain that? Well, the first thing we do, unfortunately, is dad the dad. Most of the time, we're wrong, and he is. Well, there's been a couple of cases where mom gave 2 copies of her chromosome 7. Dad gave 1. He wasn't a carrier. Then that baby had trisomy 7, which is 3 copies of 7. And they said, hey, this cell's not stable. This is not right. I'm going to lose one. And they lost the dad's. And the kid has 2 of mom's chromosomes. So that's what we call uniparental, 1 parent, disomy, 2 chromosomes. So that's very rare, but you can see that as well. And then deletions and duplications I've mentioned already, 2% of people. And then once in a really, really rare blue moon, you might just get a spontaneous blip in the CF gene to decide to make an alteration, a misspelling on its own, and we call that a de novo, out of the blue, of new mutation. Mom and dad are not carriers, and that kid still has CF. So this is just I just want to show kind of the applied genetics that we do in our clinic. The blue square here, this is what we call a pedigree. A pedigree is a family history drawing. So you have dad and mom, circles are females. They have 1, 2, 3, 4 children, okay? This child ends up to be a carrier from this parent. They marry someone who's also a carrier, and they have 1, 2, 3, 4 children. This one, the blue square, is a boy with CF. So the typical scenario we might have in our clinic is that the sibling comes to us and says, I want to know if my baby has CF. I'm pregnant, let's say. And so because ooh, I made a mistake. That should be a circle. Wow, I'm not so liberal.
San Francisco.
So, sorry, that should be a circle. I could draw it really liberal and have, sperm egg donors and all this stuff all over it, because we see that a lot too. But anyway, that should be a circle. And if you're a sibling of someone with CF, you have a 2/3 chance of being a carrier. Okay, there's 4 possibilities. You either have no CF genes, you either have CF, you're either a carrier from your mom, or you're a carrier from your dad. Well, typically by the time the person's going to have a kid, we kind of know that they don't have CF, so you rule that one out. So there's 2 out of 3 possibilities that that person's a CF carrier. So that's where the 2/3 comes from, if you can see that far. And then let's say this person marries someone who is a man or who's a Caucasian man. Their background risk will be about 1 in 25. So the risk of having being a carrier is 1 in 25 for this spouse, 2/3 for this sibling of a CF person, and then both of them have to pass that gene on to have a child with CF. So that's 1/2 and 1/2, so 1/4. And therefore that child's risk of having CF, if it's not born yet, is 1 in 300. So that's kind of how we calculate for different family members. Maybe we'll get a cousin or, a great aunt or someone, who wants to know. So that's kind of one way that we use genetics for CF. So lastly, I just want to share with you a couple of resources for those of you who are interested. There's a great database of all the different CF mutations that you can look up. It's from the Hospital for Sick Children or Sick Kids in Toronto, and there's the website for that. You can look up your mutation. They just give you kind of reports and what other people have it and what they're like health-wise. Again, you have to take everything with a grain of salt because environmental factors play a huge role. GeneReviews.org is a wonderful resource of comprehensive information from everything from treatment to diagnosis, et cetera, for CF as well as hundreds of other diseases. Online Mendelian's Inheritance of Man is a great, great way to have a sleep aid if you want to read that, because that will let you really get confused and just be overwhelmed with genetics lingo. But it is very kind of a comprehensive summary of all the different research. And then the recommended reading that I loved are these books on genetics. They're for the lay population. They're just about the genetic revolution and what's happening. And one of them is by our good old Francis Collins, Dr. Francis Collins, who co-discovered the CF gene. He wrote The Language of Life, which is an incredible, very thoughtful kind of examination of genetics in our society today. And then Dr. Ruth Cohen wrote a book called Hereditary and Hope, The Case of Genetic Screening, which is the history of genetic screening. Doesn't include CF, but it's all the other diseases that are screened for. And stay tuned, Johns Hopkins is making a wonderful website called CFTR2, which will be the American version of the CF information for the genes or for the mutations. And then I have to thank all the wonderful people who helped with this slideshow and all the slides that I kind of borrowed. And also the people in the North American Genetic Consortium who are really working hard to find modifier genes. Dr. Drum, Cutting, Knowles, there's a bunch, Zelensky, all these other people that are just dedicating their lives to help understand the CF gene, and I thank them. And thank you. And lastly, Dr. Francis Collins had this quote that I want to leave you with. All illnesses have some hereditary contribution. It's been said that genetics loads the gun and environment pulls the trigger. So remember, we are not just our genes. We are not destined to our Delta 508s. We have so much more that can help us live long, fulfilling lives with CF, regardless of what genes we're brought into this world with. So thank you very much. If you have any questions, please email me. Thank you.
I do have one question from the online audience, if you could take that. And it's just, can you comment on the news out of Philadelphia of gene therapy for sickle cell anemia as relates to CF?
Unfortunately, I don't watch TV. That is my comment. Unfortunately, I have not heard about that. I can quickly look it up, but have you heard of anything? No. As far as I understand, gene therapy for CF is not active right now. I could be incorrect, but that's my understanding, is that because there was a mishap in gene therapy with a different genetic disorder years ago, things have been kind of steady on hold. I think the real gene therapy, if you will, is the making of these new drugs. Any other thoughts, Sandra?
No, I also don't watch TV, so.
So, anything else? Okay. Thank you.
One last comment.
And I think we need another special thanks for this amazing talk. Thank you. Thank you very much. And aren't we lucky, at the end of this presentation, she even left a contact email so that those of you who think of questions as you're driving home have a place to go, and you can always contact CFRI, and we can find Anna or someone to help to answer your questions. I want to thank everyone here tonight for coming, and I want to give special thanks again to Genentech and AMED Healthcare for making it possible for us to bring the information to the people who most need it. Stay with us next January, next year. The 10th is the second Tuesday of the month, and CF Discovery Series will carry forward. We have an evaluation sheet here that we'd like you to give your feedback on, and have a good evening and a wonderful holiday. Thank you.